Good afternoon, everyone, and welcome to this final plenary session of the conference. My name is Dionne. I had the honor of being the chair of the program committee of this year's edition of NWO Physics. I hope you're all enjoying the conference. I think we really have a really great, broad and diverse set of speakers and set of sessions. And of course, we're all really happy that we're actually here again, in person, live, after 2 years of having a virtual meeting. And first of all, I would like to ask you all to really give a big round of applause to all the people that actually made that happen. That's everyone at NWO, Emma and the entire team, and of course everyone here at the Königshof. Before I start introducing our final speaker, also just a quick reminder that directly after the lecture, we have the post-award ceremony, so please stay in your seats and stay around for the prizes. Now it's my pleasure to introduce our closing speaker, Jennifer Dionne. Jennifer joins us from Stanford University, where she is the Vice Provost of Research Platforms and a Professor of Material Science and Engineering. And Jennifer is really a pioneer of controlling light and light-matter interactions at the nanoscale, and really using these new developments for new nanophotonic materials, methods, and devices. In particular today, she will show us some examples of how these really platforms, nanophotonic platforms, can be used in health applications, such as pathogen monitoring or chip-based detection of genes or proteins. Now, Jennifer has already received too many awards and prizes to list them all, so I just want to name a few. There's the Presidential Early Career Award from President Obama, the Adolf Long Medal from OSA, the New Innovator Award from the NIH, and recently the Alan T. Waterman Award from the NSF. Moreover, in 2013, she was included on Oprah's list of 50 things that will make you go wow, and I'm sure she will also make us go wow today. So please join me in welcoming Jennifer Dionne.
Thanks so much, Daniel, for the introduction, and a big thanks to the organizing committee for putting together such a wonderful conference. It's amazing that so many of the physicists in the Netherlands can come together under the same roof to share what they've discovered in the last handful of years and kind of really lay out what the future of physics might hold. It's also an honor to be here. As Daniel mentioned, my name is Jen Dionne. I'm a faculty member in Material Science and Engineering at Stanford. I also have an appointment in radiology in the School of Medicine, and I'm excited to share with you how nanophotonics is potentially enabling a new era of in situ and real-time monitoring of biological systems. And this was work that wouldn't be possible without an incredibly talented team of students and postdocs. You'll get to see many of their pictures throughout my presentation. I also want to acknowledge my collaborators, including the Director of Clinical Virology at Stanford and the Director of Clinical Microbiology, who were instrumental both in sharing their pain points when they were trying to deal with either clinical or environmental samples, and also in helping us test our devices in the real world. I know we're at the end of a long conference, and we've already had lunch and coffee and maybe aren't quite ready for our pop quiz, but I thought, what a better way to end the conference than to test our knowledge. So if we think about biology, it is an incredibly complex and data-dense system. And imagine, if you will, that we just took 1 milligram of DNA. How much data do you think is stored in that snowflake-sized amount of DNA? And you can think about each base pair as being roughly equivalent to a byte. Any guesses? The front row is looking kind of quiet. Anyone in the back? Okay, I'm hearing petabytes. I've heard megabytes. Exabytes. Spot on. So it's 1 exabyte, which is 10 to the 18 bytes of data. This is an enormous amount of data, and basically, let me go back one slide, equivalent to all of the data stored and generated by Amazon, Meta, Google, and Microsoft in a day combined. And now, why is that data important? Well, we as humans, as well as all animals and plants and other organisms, are kind of constantly shedding this data into the environment and basically leaving behind traces and clues as to how healthy or diseased we might be. And to illustrate just how powerful this biological data can be, one of my colleagues at Stanford, Colin Klosek, is actually an oceanographer and went out and collected just a few droplets of seawater off the central coast of California. And then from that seawater, he was able to extract the DNA, sequence it using a conventional Illumina DNA sequencer, and from that be able to map out ecosystem diversity just from this droplet of water. So he was able to detect over 80 different species, including sharks and whales and dolphins and seals and parrotfishes and some jawless fishes, simply by looking at the environmental DNA that got shed into the ocean water from the organisms that had passed through his site off of Monterey Bay Aquarium. However, the rate at which we can read the data that's in a biological sample is still quite slow. So we have over an exabyte of data in just a snowflake-sized amount of, say, DNA. And DNA is the fastest biomarker we can read, and we can only read it at rates of about 400 bytes per second. And if we compare that with the rate at which we receive internet data in our own homes using state-of-the-art fiber internet connections, it's orders of magnitude slower. So typical internet speeds here in the Netherlands are about 113 megabytes per second. And of course, we have proteins that we want to identify as well, and the rate at which we can read proteins is much slower, less than 1/10 of a byte per second. And then there are millions of cells and metabolites that we might want to be able to look at as well. So the talk today will present how we can use silicon photonics and essentially physics to open up rapid reading of biological systems. And first, I just wanted to share with you why DNA and protein and metabolite sequencing are so slow. And in part, it's because the reading is rate limited by chemistry. So most of the methods that are being used now are chemical methods. And to illustrate that, I just wanted to play this video by Drew Barry. He's a MacArthur Fellow who makes animations of biology, but based on the science that's out there. And this is a pretty impressive animation, in my opinion, that he made in 2010 of basically the DNA replisome. It's a pretty impressive biological motor comprised of many proteins that basically unzips double-stranded DNA into single-stranded DNA, sends it through DNA polymerase, and then takes base pairs from the environment and is able to create a second strand of double-stranded DNA from that single one strand of DNA. So basically from double-stranded DNA, unzips it into 2 single strands of DNA, and then is able to add on the base pairs at 50 bases per second to be able to essentially replicate or amplify DNA. And that's the basis for PCR and also for sequencing. Essentially, we rely on these enzymatic methods to amplify the sample. In this presentation, I want to share with you how instead of amplifying the sample and relying on these chemical or enzymatic approaches, we can instead amplify light to be able to get us much more sensitive detection. So the first part of my presentation will share some of the physics platforms that we've developed in my laboratory and in principle with the broader community who's really helped us understand how light interacts with silicon nanostructures to be able to precisely control light on the molecular scale. And then in my presentation, I'll share with you how we've used that platform for targeted DNA detection, showing you how we can functionalize the surface with probes that are specific to, say, certain gene fragments that we might want to detect in environmental samples. And then I'll also share with you how we can use the platform for label-free detection. In principle, how we can detect millions of different types of cell species and strains and their antibiotic or drug susceptibility by relying on vibrational fingerprinting of those biomarkers. And then finally, at the very end, I'll share with you how we're starting to deploy these systems now on autonomous underwater vehicles and in various communities to be able to do wastewater detection to detect pathogens in the wastewater. And really, our goal as a lab is to be able to better inform models of health and disease of the environment, essentially by using these in situ and real-time sensors, and then also to switch from a more reactive and crisis-based approach to human health to basically a more proactive and solutions-based approach. So many of you throughout the pandemic have probably had the chance to use one of these sensors at home, basically a rapid antigen sensor. We know it's miniaturized, and we know it's based on a color change. And that color change basically comes from aggregation of gold nanoparticles. So you've probably seen beautiful rose windows or stained glass windows. Those are basically metal nanoparticles that are doped into the glass, and they have very vivid optical scattering. So the color change of these sensors is very similar to the physics of what you would find in rose windows or stained glass windows. Essentially, there's a layer of nanoparticles that is functionalized with an antigen. And when you have the SARS-CoV-2 protein, it will bind to the protein that is already in the sample, along with another nanoparticle, and cause aggregation of those nanoparticles, which gives rise to a vivid color change. The problem with these sorts of sensors that we've all seen is that they are great for rapid at-home tests, but they have relatively limited sensitivity. And that comes from the broad scattering spectrum of these metal-like nanoparticle sensors. So if you look at the scattering intensity as a function of wavelength, you can see there's a pretty broad full width at half max. So if you're trying to sense just a single antigen, like the receptor binding domain spike protein that's on the SARS-CoV-2 virus, there's just a small shift in the resonant wavelength that turns out to be pretty hard by eye to detect. And then this continues on until eventually you get a large enough shift with a high enough number of molecules that is bound to the metal nanoparticle. This talk will focus on high-quality factor resonators that essentially allow light to be stored in the device for much longer periods of time, giving rise to much sharper resonances and much sharper full widths at half max. And in this way, we can get sensitivity down to the few to single molecule level, because now you can see there's a much more distinct change in the intensity when there's just one or a few molecules being added onto our sensor. So we rely on silicon, and like I said earlier, the motivation was trying to use a lot of the silicon photonics industry and the speed with which we can get fiber optical communications to be able to more quickly read out biological data. But what we need to do is basically shrink down light, which has a wavelength of anywhere from 400 to 700 nanometers at visible wavelengths, to the size of a molecule. So what we do is develop nano antennas that are able to concentrate light down to very small volumes, but also store light for a substantial period of time, so that way we can get those very high quality factor resonators. And the structure I'll present to you today is shown here. It's essentially a silicon on glass chip, so it looks a lot like a conventional microscope slide that you might use for any biological imaging. And it's comprised of these nano antennas that look a little bit like dog bones. And then we broadly illuminate the chip from the bottom, and then essentially take images from the top. So you can see that each nano antenna, each dog bone, has a dipole-like scattering, so that way there's a one-to-one correspondence between each nano antenna and each pixel that is on our detector array. And here you can see the images on the CCD camera of what the scattering looks like from each of the nano antennas. And then just to talk you through the physics of the nano antennas, since we're all physicists in the audience, there's essentially a cavity region which is responsible for concentrating light and storing it there for extended periods of time. And then we also have a photonic crystal at the end that basically further traps light in this cavity. And then what we can do is flow in our sample, and based upon target binding to any of the nano antennas, or based upon how the spectra changes, we can detect what's landed on each nano antenna. So to describe in a little bit more detail how these silicon nano antennas work, let's consider just an array of silicon bars that are all the same width, and let's pretend we're illuminating it from the top. So in this case, we're illuminating it basically at normal incidence with respect to the screen. And what you can see is that the electric field of light basically is oscillating between +1 and -1 in these silicon bars that are just about 100 nanometers across. And if you look at the scattering spectrum, so the light that gets scattered back to our eyes, it's pretty nondescript. What you'd wind up seeing are a bunch of Fabry-Perot resonances, all of which have pretty broad full widths at half max. Now though, if we consider subtly changing the width of neighboring bars, we take an electromagnetic mode that normally would have been forbidden or completely dark and make it leak a little bit of light out. So you can see just by changing the width of these bars a little bit, we're able to trap light in the structure and enhance the electric field, now over 150 times the initial magnitude. So this structure is essentially acting like a nano antenna, and now when you look at the scattering intensity, you get this extremely sharp peak right around about 15, 20 or so nanometers. And there's a zoom in, and the inset is essentially a phonoresonance between a waveguide mode, that's along the length of the bars, and kind of a localized resonant mode within each of these silicon bars. And if you plot the quality factor of this structure as a function of the difference in width between each structure, it keeps going up. So to tens of thousands to millions based upon the difference between each neighboring bar. And the smaller that difference, the higher the quality factor will be. The thing is, in those simulations that I was showing you, we assumed that the waveguide was essentially infinitely long. And if we want to have dense multiplexing on a silicon chip, we need to be able to shrink down the size of these nano antennas so that we can detect many biomarkers simultaneously. The thing is, as you start truncating down the waveguide, and again, here we're illuminating it from the top, you can see now there's scattering out of the edges of the waveguide. And that scattering is going to reduce the quality factor of our structures. And here you can see calculations of how that quality factor basically dives down as your nanocavity length gets shrunk down from essentially infinitely long or semi-infinitely long to below about 10 microns. So the trick that we use is basically to put mirrors on the ends of the cavity so that way it seems like light is propagating, somewhat out to infinity, even though it's confined in a smaller cavity region. And if you think about how to design a proper mirror, we can take a lot of inspiration from the photonic crystal community. And basically, any energy that lies in the mirror's band gap, or in the photonic crystal band gap, is going to get reflected. And you can think about that just from a simple solid-state physics point of view, or materials physics point of view. If you have a material that has a band gap, and you try to send in light at that band gap, if it's below the band gap, it's not going to get absorbed. You don't have enough energy to absorb, say, electrons to the conduction band, so it's just going to get reflected. It's very similar for these photonic cavities. And the strength of the reflection is based both upon the lower edge and the upper edge of that photonic crystal band gap, as well as the mode frequency that you're sending into the photonic crystal. So we want to have a mirror that's quite reflective, so that way light can be confined in a finite length structure, but we don't want it to have such a high impedance match that basically there's a lot of light that leaks out of it. And there's this really beautiful paper by Noda and colleagues published now over 20 years ago where they have the quote, "You want to confine light gently to confine light strongly." So you don't want to have such a strong impedance match that light scatters out of it. You want light to be adiabatically kind of propagated into that photonic crystal mirror region, so that way it can be strongly confined and basically remain within basically have no mode outside of the leaky region. So what we do is basically model the cavity and also the photonic crystal mirrors, determining what their band gap energies are and what their dispersion diagrams are, what their band structures are. And here on the right-hand side, I'm showing you the band structure of the cavity itself. We're going to be most interested in this mode that's just below about 220 terahertz. It's in the near infrared. You can see it's located at a normalized wave vector of 0, so that means it's going to be propagating to the detector at normal incidence. And then what we do is also calculate the band structure of the photonic crystal mirror that's going to be on each end of the cavity. And what we want to do is essentially have that mode in our cavity at 220 terahertz be in the center, basically, of the band gap of the photonic crystal, so that way we have maximum reflectivity. So what we do is design these photonic crystal mirrors where we vary basically the width of the block to optimize the reflectivity while still confining light gently yet strongly. And we can do that by varying the taper region. And you can see that the quality factor now in this structure that's only 8 microns long has been boosted from just a few hundred now up to over thousands and tens of thousands, at least in theory. And you can also see where light gets strongly confined in that cavity region of this dog bone-like structure. We can fabricate these using standard photolithography or electron beam lithography in Stanford's clean room. And what we can do is measure what the quality factors are when they're in physiological buffer, and we have extremely good agreement between what our theory predicts the quality factor should be and experimentally what we get. And in addition to those high quality factors that allow for very sensitive molecular detection, as I showed you, these structures also focus light to a rather extreme scale. So for some of the cellular-based applications that I'll share with you today, we're able to use that dog bone structure where most of the light gets reflected just back and forth in that cavity that's a few microns long, and that's comparable to the length scale of a typical eukaryotic or prokaryotic cell. And then what we can do is further engineering where we introduce, for example, slot waveguide structures into this resonator to be able to shrink light down to attoliter scale volume, so truly to the single molecule regime. And here you can see the mode volume as a function of the device length. You can see with certain structures, we're able to shrink the mode volume down to less than 1/10 of the wavelength of light, even though we're broadly illuminating many of these resonators simultaneously. And importantly, we're doing so at a wavelength where silicon is transparent, so we're not heating up the cells or the molecules as we're interrogating them. So with this platform, we can pattern many sensors on a chip. A 1 centimeter square chip that we patterned in the clean room can contain over 3 million sensors. And for those of you who are maybe biophysicists or who have ever worked in a microbiology lab, this is equivalent to over 31,096 well plates that you would use for an equivalent number of, say, proteomic assays in a biology lab. So if you were to use 31,096 well plates, you'd basically need about the size of a basketball court to do an equivalent number of assays that this silicon structure allows for just on a 1 centimeter square area. And then these chips are especially useful because what we can do is put down unique chemical functionalization over each biosensor. So I heard some pretty impressive talks on the physics of fluids and saw some really amazing posters there, so I wanted to include some of this data where I've collaborated with Professor Pierre Kuryakov in electrical engineering, who's a pioneer of acoustic droplet ejection, and essentially a lot of the printers that you used to use at home, like the inkjet printers. And he and I were able to basically adapt those inkjet printers, but make them be able to print biological reagents, but in very small volumes. So we use basically an acoustic wave via a piezoelectric transducer to print droplets that are just a few microns in diameter, and then they can basically deliver reagents over individual regions of the chip. So based upon the piezoelectric frequency that we're using to generate the acoustic wave, we can create different droplet sizes down to below the 10 micron scale. And then here's just an example where we patterned the Stanford logo over each of our individual resonators, here with fluorophores that are in the red and in the green. In addition, for kind of field deployability, you don't always want to have to work with a spectrometer, so what we can do is essentially hyperspectral imaging with like a tunable series of lenses. And here what we're doing is illuminating the chip using a pretty low resolution camera, and you can see each sensor now is lighting up. So there's no fluorophore attached to it. We're essentially just imaging the light that is being transmitted by each of these dipole scatters, and you can see they're lighting up at different wavelengths based on what is bound to the chip. So here each frame corresponds to a distinct wavelength, and that allows us to build up the spectra even if we're not using a spectrometer. And you'll also notice that here, even though this is silicon on glass, basically these antennas are so bright that the glass background is essentially dark compared to what each of these nano antennas is sending to the detector. So I'll summarize the physics so far of these sensors. And to summarize it, I like to think about two orthogonal axes that I think are very useful for biosensing. One of which is the spatial or diffractive control of each of these nano antennas. Essentially, how good are they acting like dipole scatters to report what's happening in the near field of the sensor to the detector. So basically, you want to have that one-to-one correspondence between what's happening at each pixel or each sensor and what's happening at each pixel on the detector. And we know that plasmonic structures or dielectric resonators are really outstanding dipole-like resonators, but generally they have low quality factors, and that is basically similar to that at-home COVID test that I mentioned a couple of slides ago. The corresponding axis that's really important, especially if you want to do sensing at the single cell to single molecule level, is the quality factor. And there are structures like photonic crystal defect cavities or ring resonators that can get quality factors into the millions or billions. So they're exquisitely sensitive and really beautiful, but it's very challenging to do multiplex detection because they don't have that controlled far-field dipole-like scattering. So this sensor platform that is essentially just silicon on glass allows for simultaneous control of both the dipole-like scattering, so that spatial control, which gets us multiplexing, but also high enough quality factors where we can do single cell to few to single molecule detection. And these structures, as you saw in the images, have a very light, flexible form factor. So we're doing everything in silicon, and I think there have been amazing advances in silicon metasurfaces, some of which we've heard at the conference today, that are allowing us to shrink down normally bulky lenses and other optical elements to thinner than the width of a human hair. So these structures have that same metasurface, ultra-thin, lightweight form factor, and they're also amenable with large area fabrication. And of course, here we're at the home of AMSL, so we can tap into the existing industry for CMOS-compatible processing. So now let's put these metasurfaces to work. And I want to share with you a couple examples. First, I'll focus on label techniques, and in particular focusing on how we can label each of the nanoantennas with DNA that is complementary to the gene fragments we want to detect. And then I'll shift to label-free techniques and how we can essentially use the vibrational scattering spectra of molecules and cells to identify what has landed on the chip, even if we haven't necessarily targeted that particular biomolecule. So the label techniques are kind of the easier ones to understand. Essentially, you can imagine if you wanted to detect specific gene fragments, for example, in ocean water, if you want to detect those 80 species that Colin was able to detect, or if you want to look at specific RNA fragments of the SARS-CoV-2 virus, what you can do is put down complementary single-stranded DNA onto your metasurface sensor, and it basically sense the complementary DNA or RNA. So we do that using self-assembled monolayers, starting with silentization of the surface. Then we put a bifunctional crosslinker on top, and then a thiolated DNA probe that's complementary to the nucleic acid that we want to detect. And then this particular paper, we're focusing on looking at the open reading frame gene fragment of SARS-CoV-2, as well as the E gene of SARS-CoV-2. And here, essentially, we just have a high-quality factor resonance that acts a lot like that at-home test, except now the full width at half-max is considerably sharper, so that we can do much more sensitive detection. So you can see with each self-assembled monolayer that gets added onto the surface, we have a nice resonant wavelength shift that agrees quite well with what we simulate based on how these structures should work. And then if we look at the total wavelength shift, keep in mind I showed you those images where we can interrogate many hundreds to thousands of sensors simultaneously. So essentially each dot that you're seeing on this wavelength shift corresponds to a distinct sensor that we're probing simultaneously to see what has bound to the sensor. So you can see as we add on different self-assembled monolayers, there's a nice roughly about half nanometer shift that we see between having the probe that is complementary to the open reading frame target and the target once it's actually bound. What we do is now construct flow cells, and they're pretty basic flow cells. So here you can see the chip, and you can see about 2 pads of 10 sensors. Each of those 10 boxes contains about 10,000 individual resonators in it that have been individually functionalized with different SARS-CoV-2 gene fragments. And then we can introduce either nasopharyngeal swabs or environmental samples to be able to detect either the environmental DNA or RNA or the SARS-CoV-2 gene fragments. And you can see here that when we're measuring hundreds of resonators simultaneously, when we add in the target, we can see a resonant shift within 5 minutes of adding the target. So it's a fairly fast readout of what's happening in the biological sample. And here we did it in clinical samples, so clinical nasopharyngeal eluids, where we introduced basically the patient sample into our resonator. When we have a sample that we know is negative, we get a little bit of a wavelength shift that comes from some non-specific binding across roughly the hundreds of resonators that were measured. Then we flow in a buffer, which kind of cleans off the sample, and basically anything that's non-specifically bound gets rinsed away. And then we introduce the samples that were tested by PCR to be positive. Now we get that nice 0.5 nanometer wavelength shift. When you flow in the buffer, the shift remains. And if we optimize the probe that's on the surface to make sure that there are no hairpin loops that are formed, basically at room temperature and pressure, we can get a lower limit of detection of about 8 femtomolar. So we have pretty exquisite sensitivity, at least with this first device design. And to put that in perspective, PCR has a sensitivity of roughly 500 to 1,000 copies per milliliter, but it takes about 2 to 4 hours to be able to replicate or amplify the DNA itself. And there's also possible inhibition, so it's actually really challenging to do this in environmental samples or like wastewater samples. Whereas our device has a worse sensitivity, so about 10x worse than PCR, but still orders of magnitude better than an at-home antigen assay, and can give results in 5 minutes without having inhibition. So essentially, by relying on how we amplify light or concentrate light on the nanoscale, we can overcome these chemical or enzymatic methods to amplify or replicate the sample. So now I want to shift gears to some of those label-free scattering measurements that I promised you. And there are many examples where you don't necessarily want to put down a probe that's specific to the biomarker you want to detect. You kind of want to do an unbiased scan of what might be in your sample. And this is an approach that we call VISMO, or vibrational spectroscopy with metasurface optics. Here we're relying on those same high-quality factor resonators, except that instead of looking at shifts in the wavelength, what we're doing is relying on how the incident light gets scattered by the molecular vibrations to longer wavelengths. So essentially the molecular vibrations of a sample give rise to inelastic photon scattering that provides a very unique fingerprint of the sample we're going to look at. And essentially we'll rely on Raman spectroscopy, and in particular looking at the Stokes shift. So we put that high quality factor resonance basically at the wavelength that we're going to be using to excite our sample, and then we also design the structures to have kind of a broader resonance. In this case, it's essentially a magnetic dipolar resonance, basically to correspond with the fingerprint region where we expect the biomarkers to have most of their vibrations. So for those of you who haven't taken inorganic chemistry in a while, I just wanted to share with you how Raman scattering is working. So we're coming in with a monochromatic light source, and then the molecular vibrations are basically adding to or subtracting from the energy of that incident light. And like I said, we're going to be looking at the red-shifted wavelengths, or the lower energies. And usually we'll plot the axis on the Raman as a Raman shift. So that basically is an absolute wave number shift compared to the incident wavelength of light. And for the most part, we're exciting at about 1064 nanometers. So the Raman shifted wavelengths are going to be roughly around 1100 nanometers up to about 1300 nanometers. So when we use the silicon platform for Raman spectroscopy, we can now get extremely sensitive detection of molecular monolayers, and in this case we started looking at peptides and proteins. So here you can see when we put proteins on the sample, for example streptavidin or an albumin protein, we don't get any Raman signal when we're off the resonance of any of the nanoantennas. But once we're matched to the resonance of the nanoantenna, here shown in that black line, you can see we start to be able to get Raman signature from just 20 or so attograms of sample that are on our surface. And what we can do now is start to use this to identify various proteins and peptides. So what we can do is look at the vibrational scattering signature of amino acids, which are basically the building blocks of proteins. And each of the vibrational spectra of amino acids is very differentiable. Here, and I'll share with you in a couple of slides how we process the spectra, but essentially using ML, or machine learning algorithms, we can project the spectra onto a lower dimensional space. In this case, it's essentially almost like a principal component analysis. It's a nonlinear one, though, called a T-SNE. And you can see each amino acid has its own unique kind of T-SNE cluster. So each amino acid is very differentiable. We can also look at peptide sequences. So here, these are 4 amino acid long peptides. Here's like an arginine at one end and a cysteine at the other end. Here's an arginine at one end and a serine at the other end. And you can see these 2 peptide sequences differ only by 1 atom. So basically there's a hydroxy group versus a thiol group. And again, the Raman scattering signature is very differentiable. We can also look at small post-translational modifications or molecules that are added on to each of these amino acids, and those also provide very differentiable scattering signatures. So it's neat when we think about the building blocks of life, basically their molecular vibrations are so distinct and they're so tuned for their biological function that we wind up being able to get very good or very high fidelity discrimination of these different molecular building blocks. So kind of armed with that foundation, we wanted to know, could we look at something as complicated as a cell? And you think about a cell as basically a balloon or a bag of like water, plus a lot of proteins and a lot of lipids. So it's a very complicated signal. We wanted to know if we send in a laser to a single cell and we look at the Raman fingerprint that comes back, can we use that to identify the cell species and the cell strains? And you might say, well, why is this important? Well, what we were motivated by is more rapid identification of bacterial infections and also their drug susceptibility. And of course, for the past 2 years, we've been in lockdown thanks to a viral infection. So I kind of view bacteria as the underdogs of the media. But yet, if we look to kind of the global population, roughly about 30% of all deaths in developing countries are due to bacterial infections. There are more than 10 million cases of tuberculosis that emerge each year. And then also by the year 2050, the World Health Organization predicts that antimicrobial resistance will be the leading cause of death, with also about 10 million deaths per year due to these antibiotic-resistant infections. So what we wanted to do is basically try to address some of these challenges by enabling more rapid detection of whether or not you have an antibiotic or a bacterial infection, and then also determine which antibiotic it would respond to. And part of the reason it's so challenging to quickly detect bacterial infections is because you normally have to wait for the bacteria to basically divide and multiply. So you have to culture the cells. And just like some of those chemical methods I mentioned at the beginning, this is just a naturally slow process that takes biological time frames to be able to give meaningful results. So at Stanford, with our collaborators who work in clinical microbiology, detection of, say, a bacterial bloodstream infection can take roughly about 24 hours because you have to wait for the cells to be cultured. And then antibiotic susceptibility testing takes an additional 24 hours. So once you've got the culture, then you need to add antibiotics and figure out which bacteria are dying versus which are still surviving. And then with TB, culturing can actually take over 40 days. It's just such a slow-growing pathogen that it takes a while to culture it to be able to do the antibiotic susceptibility testing. And then in the meantime, you're either a patient in the hospital or you're a person out with an active TB infection. And according to the CDC, over 50% of patients are given antibiotics when they don't need it. And that's because the risk of not having antibiotics is so high if you do have a bacterial infection. But of course, this is giving rise to antimicrobial resistance. And I was just chatting with some folks over lunch who actually mentioned in the Netherlands it's quite challenging to get antibiotics because the EU is so concerned about the rising cases of antimicrobial resistance. So bacteria can be engineered to have certain colors, but in their natural state, they're colorless. If you've ever cultured bacteria, for the most part, they look kind of whitish or yellowish. So we wanted to know, could we use Raman to essentially distinguish the color, so to speak, of bacteria that haven't been engineered to have any sort of fluorescence or absorption? So we started by looking at over 31 bacterial species and strains that represent over 95% of all cases seen at Stanford's hospital, including E. coli, Pseudomonas pneumonia, MRSA, MSSA, strep, as well as some yeast samples. And we collected many single-cell Raman spectra. And then what we did is basically develop a neural network where we could feed our spectra into a neural network to train it and basically say, whenever you see this sort of Raman spectra, you know that that Raman spectra, say, corresponds to E. coli, because we have the ground truth of what E. coli looks like from molecular methods or from mass spectrometry. And then we give it a spectra that the neural net hasn't seen before and say, hey, what cell is this most like? And the neural network outputs a series of predictive scores that say, well, it's probably most likely E. coli, or maybe it's most likely Streptococcus. So what we did is train this neural network on over 60,000 spectra that correspond to individual cells, and then we tested it on over 12,000 spectra. And you can see that the neural network actually does quite good. So we can give it just a 1-second Raman spectra of a single cell, and it can spit out basically which class it most likely responds to with pretty high accuracy. And if we zoom out to the full 31 species and strains that we have looked at this point, we can see that the average strain level accuracy is about 82.4%. But more importantly, you can see here each of the colors corresponds to that same first choice antibiotic that a clinician would use based on empiric evidence. And we actually get an average first choice antibiotic treatment accuracy of over 97%. So comparable to mass spectrometry, but in this case without having to necessarily do the culturing. Those were on cell line samples. We also wanted to see if this translated to clinical samples, where we focus on clinical bacterial bloodstream infections where the patient had tested positive, and we want to see if we could identify the bacteria that was in the blood. So here you can see as we started adding in 30 patients and basically training the data on some of the clinical samples, we were able to get, in most cases, accurate prediction of which antibiotic should be used. And then as we added in more patient samples, so here an additional 30 patient samples, the neural network was getting more and more accurate. So essentially the more examples of clinical data it saw, the more highly the result would predict correctly the antibiotic treatment. And importantly, we only need about 1 to 10 cells to be able to get accurate classification of the bacterial species and strains. So you don't need to wait for an overnight culture. You can just have a few minutes of a culture to be able to correctly predict at least what the first choice empiric antibiotic would be. Then we wanted to know for a more challenging bacteria like tuberculosis, where it normally takes 40 days to culture, can we determine its antibiotic susceptibility? So what we did is collect samples of TB. In this case, it's a cow strain of TB, so it's a little less dangerous to work with. But we had TB that we knew was resistant to 4 different antibiotics, so amikacin, moxiflacin, isoniazid, and rifampicin, as well as the wild-type tuberculosis. And then again, we collected Raman spectra, and here we collected Raman spectra for about 15 seconds. And then we fed that into our neural network, so basically trained it on TB strains that we knew were resistant to certain antibiotics. And then on spectra the neural network hadn't seen before said, what do you predict is the antibiotic resistance or susceptibility of this spectrum? And again, you can see really nice groupings in our T-SNE that show that we should get highly accurate prediction of which antibiotic the TB is resistant to. And what's interesting is what we can also do with our neural networks is remove certain wave number features and see how the accuracy of the algorithm drops down when you remove certain wave numbers. And for tuberculosis, it's known that resistance comes from basically a thicker mycolic cell wall, or basically resistance corresponds to mycolic acid building up in the TB. And we saw that the wave numbers where the accuracy dropped down the most when we removed them actually correspond to those mycolic acid vibrations. So looking at the output of the neural network, you can see we get over 98% accuracy with just 15-second acquisitions of TB. We can also detect different genetic variants. So out in the wild, different clinical samples will have different point mutations in the DNA. So even though all the TB, say, will be resistant to amikacin or resistant to isoniazid, there will be different genetic mutations. And what's interesting is that we can still determine, even with those genetic mutations, which drug the TB will be resistant to. And we can also get reasonable accuracy to say what the genetic mutation might be, what that single point mutation might be. And then I showed you that we can develop ML algorithms that basically allow us to understand which features, which wave numbers are most important for TB classification. And you can see that basically we only really need about 100 or so spectral features to be able to get very high classification accuracy. So So this goes back to at the beginning where I mentioned you don't always want to have a spectrometer, right? That can be an expensive and a bulky component. Here what we can do is just hone in on those features that we know are most important for classification accuracy, and then develop hardware that's much more miniaturized and low cost. So here's my student, Baba, who developed, along with Manu Prakash, essentially a handheld Raman spectrometer that doesn't rely on spectroscopy. It just has a series of filters. And now in basically sputum samples, what we can do is image each TB cell that's in a sputum sample and then determine with very high accuracy using this very low cost and portable Raman spectrometer which antibiotic it would be resistant to in just 15 seconds. So you no longer, in principle, would have to wait the 40 days to be able to do the culturing and the antibiotic susceptibility testing. So in the final few minutes, I want to share with you how we're now developing this for wastewater-based epidemiology and kind of population health monitoring. And at least in the US, and I imagine here in the EU, we've seen how PCR has enabled the detection of viruses in wastewater, so that way you can monitor, for example, the prevalence of SARS-CoV-2. It turns out it's actually quite a bit more challenging to be able to monitor bacterial infections and bacterial drug resistance in wastewater, in part because there are so many different gene fragments you want to look for the dozens of bacteria that could be present. And in addition, like I mentioned, PCR suffers from inhibition. So basically those enzymes don't work when there are a bunch of other salts and waste that might be around. So again, the idea was to use Raman spectroscopy, and in this particular case, we just took wastewater, which had spiked bacteria in it with known concentrations, here down to about 8 times 10 to the 6 cells per mil, which is a typical concentration of bacteria that you find in wastewater. And in this case, we just used gold nanoparticles. And we're able, even in wastewater, with Raman spectroscopy, to just shine the laser in and detect that there are E. coli or staph or even mixtures of bacteria in the wastewater. And I think this is neat because we're not necessarily now getting signal from a single cell, and in some cases the concentrations are so low where our laser might not even be illuminating that cell, but I think it's leaching out basically metabolites that are giving us an overall fingerprint or idea of what bacteria are in the wastewater. So I'll just end with a couple slides of what I think the future holds with these nanophotonic platforms for health monitoring. In many of the cases I've shared with you, we've either detected a single type of biomarker or certain gene fragments, for example, DNA or proteins or metabolites. But because we can fit so many sensors on a single chip, we have the opportunity to do multi-analyte or multi-omic detection, which I think is really cool, especially if we want to learn more about the environment, and in particular, who is doing what under which physical conditions. And one of my postdocs, for example, is really excited to develop this for monitoring harmful algae blooms, where you want to know not only what the population of cyanobacteria is, for example, in the ocean, but you also want to know how that's affecting the overall ecosystem health by monitoring the environmental DNA, and to what extent a toxic metabolite, in this case microcystin, is also entering the environment. And to that end, we've also started collaborating with Monterey Bay Aquarium and the National Oceanographic and Atmospheric Administration to be able to develop these miniaturized sensors and put them on autonomous underwater vehicles. So you can imagine that sputum or nasopharyngeal swabs or blood samples are pretty complex to work with. Wastewater and ocean water are actually quite a bit more complex because there's so much debris in them. But my collaborator Chris Sholin at NOAA and also at Monterey Bay Aquarium has developed this environmental sample processor that can essentially take liters of ocean water and filter it on board the autonomous underwater vehicle, so that way it can go into our chips and basically read out dynamically what the ecosystem, environmental DNA or proteins in cells, are doing as a function of time. So I'll wrap up by saying hopefully this talk has given you a flavor of how nanophotonics, and in particular the physics of silicon nanostructures, can really allow microbiology to give us insight, real-time insight, and high data insight into ecosystem health. So I started off sharing the platform itself, these high-quality factor metasurfaces that allow for very tight confinement of light and also high addressability, so we can look at many hundreds, thousands, if not millions of sensors simultaneously. And then I shared with you targeted detection, so we can use acoustic bioprinting to put down different functionalizations at different areas of the chip, and then detect specific gene fragments. And then I also shared with you how we can use label-free Raman spectroscopy to detect bacterial species and strains and their antibiotic susceptibility. And in the future, I'm really excited for how physics is essentially opening up a new toolbox for us to understand the mysteries of biology. For example, how drugs and pathogens might be interacting, and also basically to develop kind of CubeSats of the sea to better inform ecosystem health and transitions from wellness. So with that, I'd like to thank the students and postdocs and collaborators who made this work possible. I'd also like to thank our funders, and a huge thanks actually to the Netherlands physics community. I had the chance to do some of my graduate work in the Netherlands when I was working at AMOF, and it's great to see so many familiar faces and folks who really have taught me how to do physics in the best possible way. So thanks so much for the opportunity to be here.
Thank you, Professor Dionne and Jennifer, for this highly elucidating talk on your research. And we're very happy that you came all the way from Stanford to talk to us about this, showing us that your research is not just interesting and important, but really has an impact on world health as well. So on behalf of the organization, we would like to offer you a small gift, which is a card game on elementary particle physics. So there you go. Everybody, please applause for Jennifer. Thank you very much.